And this is where we left off in the last one. And just to reiterate that carbohydrates have a wide range, a wide variety with regard to their arrangement. If you think back to chapter 4 and the different ways that the carbon bonds can be created but still be a carbohydrate. And that makes them very, very useful for cell recognition, that each cell type can not only be typed for the species, but also what kind of cell it is. Now, we talked a little bit about how a protein is made, and we talked about this concept, how the protein is made, directed by the nucleus, moves out of the nuclear envelope to the endoplasmic reticulum to the Golgi apparatus. And definitely in the Golgi apparatus, we talked about the cis and trans, so the sidedness of the Golgi apparatus. But actually this is true all the way along, is that you have sidedness of each of these membranes. You also have an inside and an outside with regard to the plasma membrane. It's fluid, but you still have the inside and the outside. And what happens here in terms of this concept of sidedness, you have these transmembrane proteins. They are created. Remember, they are dictated by DNA, created via the endoplasmic reticulum, then packaged in a vesicle, which is sided from the endoplasmic reticulum to the Golgi apparatus. Golgi apparatus then repackages them. Maybe you'll have a protein, you'll have a glycoprotein, you'll have a lipid and a glycolipid. So you'll have various things, and in your book what you have is the thing they're giving you is a protein, actual protein that is a purple thing, and then you have a barbell, and then you have the barbell attached to the protein. So you have these simpler ideas, and then they are going to be repackaged maybe together into a vesicle in the Golgi apparatus, but you still have this concept of sidedness, where in this picture anyway, just this picture, outside of the membrane is that orangey tan color, inside is kind of the purple or the turquoise color, sorry, not colorblind. And you have this in your book also. So what happens is, as these proteins move toward the cell membrane, you still have this idea, and it actually the cell membrane in a way, as you can think of it, as almost opposite, because its outside is the same as the Golgi's inside. So what that vesicle does is then comes close to the plasma membrane, actually integrates with the plasma membrane, and then opens up and becomes part of the plasma membrane. So that vesicle that the Golgi made becomes part of the plasma membrane, and those proteins or those carbohydrate markers that were actually attached on the inside of the vesicle then become part of the cell membrane. And so you have this capacity for the DNA, for this cell itself, to direct changes in the plasma membrane. And these changes can be because now there's suddenly a tremendous amount of some sort of chemical the cell wants, and now we want to have a channel for that particular, maybe it's sodium, maybe it's an ion channel, maybe it's a particular protein that can't get in any other way, but we want to have something to facilitate the ability for that to get in the cell. And you have this capacity in the fact that the vesicle is made with that protein or carbohydrate attached to it on the inside, and then it becomes one with the plasma membrane. Very interesting. In terms of permeability, the cell exchanges material with its outside surroundings. Some things can pass through, some things not. So some things pass through in a passive way. So you think of gases and some hydrophobic molecules, some molecules that can pass through So hydrophobic molecules such as hydrocarbons can dissolve into the lipid layer and pass through. And some large molecules pass through slower. They just take protein channels. So they pass through slower or take protein channels. Some actually have to have their own channels to pass through. So there is a variety of ways that the cells can take in the nutrients or take in the chemicals that they need. A little bit more about transport proteins. They can be specific, so they can be just designed to take one particular chemical in. I think of sodium. Sodium is Na+. Sodium is very, very important for a cell to for nerve impulses. So the types of proteins can be very, very specific, or they can just be non-specific channels. So they can be non-specific channels that allow anything that is the proper size or the proper charge to pass through. Transport can be passive or active. And so now we're moving on to kind of a different topic, so kind of rearrange your head a little bit, I guess. So transport can be passive, or or the cell can use some of its well earned or well used or well made ATP to actually bring something in. So it can be active transport, but passive transport doesn't require energy on the part of the cell. All chemicals have a kind of thermal energy. That thermal energy is because they're constantly moving away from each other. That thermal energy allows for there to be a passive movement of chemicals, and that's what we talk about passive transport. The most common passive transport is diffusion, and we define diffusion as the passive movement of molecules from a higher concentration gradient to a lower concentration gradient, otherwise known as down a concentration gradient. And you can think about if you have a glass of water and you drop a drop of red food dye in that water, the drop will stay in the middle for just a little bit, and then the dye will diffuse out, will go out to all parts of the water until eventually the water becomes all the same color. So that drop of dye becomes totally confused, totally not confused, hopefully it's not confused, diffuses throughout the entire water. And that is from a high concentration gradient to a lower one. That's what's happening until it reaches equilibrium. And we see this in the top image, the molecules of dye. We've dropped them in water. They're the orange dots in the top image. They are going to distribute themselves throughout the water until they are equally distributed in all areas of the water. So you have equilibrium there. This also could happen if you put two drops of dye in, so you put a blue one and a red one in, you have a very high concentration of blue on one side and red on the other, but eventually you're going to have purple. You're going to have total diffusion of all the molecules in throughout the whole area. So on the bottom one you have all of the orange dots on one side and the purple on the other, and what they're going to do is they're going to continually move to equilibrium until you have a distribution of orange and purple throughout the whole area there. There is another type of a specific type of diffusion, that is diffusion across a semi-permeable membrane, and that probably sounds pretty familiar to you, and it's particularly water diffusing across a semi-permeable membrane. We call this osmosis. And so the bottom written portion of this slide, osmosis is the diffusion of water across a differentially permeable membrane due to free water concentration. And we'll look at an image on the next slide about that. Here you have the idea of osmosis across that selectively permeable membrane. And what happens is the water can pass through the membrane, because that's what the selectively permeable portion of this comment means. So the water can pass through, but the solutes can't. So what happens is you're going to end up with more water in one area than another. And the reason that takes place is because there needs to be an equilibrium in the amount of solutes, not the water. The water is what actually can pass through the membrane. So the water can pass through the membrane so that that can then equalize the concentration of solutes in each of the compartments. And this should sound somewhat familiar, because that's what happens in the cell. You have water outside the cell and inside the cell, and the concentration of solutes needs to remain in an optimal level in order for that water to remain in what is homeostasis on the outside and inside. And what we're looking at with the rest of this image that you see here on this slide is what happens if in fact that doesn't take place. We tend to like to live in an isotonic solution. Isotonic, the solution concentration is the same on the inside and outside. So there's no perceptible net movement of water. Now, don't get me wrong, water moves through the cell and outside of the cell, but it stays in equilibrium. It's constant, it's moving in as much as it's moving out. So that equilibrium is constant, with great adaptation, great movement, keeping it constant, I guess I might say. In a hypertonic solution, you have solute concentration is lower inside the cell than outside. So hypertonic solution. So you drop the cell in a solution that has more solutes in what you've dropped it into than what's in the cell is going to lose water and shrink, and that's what you're seeing in the in the middle row. You're seeing that in the animal cell, it's actually a blood cell that is going to shrink and shrivel up. Hypotonic solution concentration is higher inside the cell than outside, and it's going to be the farthest to the right in the red blood cell, and so that's going to cause that cell to actually take in water to the point where it actually break open, it'll burst. Osmoregulation is the control of water concentration, and this is absolutely, I mean, you can look at the pictures of the cells, red blood cells. We don't want our red blood cells to shrink to nothing, nor do we want them to burst open, and so it's absolutely important for life to continue, so it's a necessary adaptation for life. The top the top row of images is where we are introducing you to these terms. Hypertonic, concentrated solutes outside, more so than inside, and you have on this particular image, inside the cell is the bluer color, outside is the oranger color, and what you have is water moving back and forth. And so hypertonic water is going to move out of inside the cell to where the solute concentration is highest to equalize them. On the third one over, because isotonic you have constant movement, the hypotonic water is going to move into the cell because the concentration of solutes inside the cell more so than outside. And this is true with plant cells also, where you get to the point where the plant cells will shrivel up. That's when your plant shows you it needs water, or in the middle you have a nice full vacuole. In the far right of the image you have the plant cell that's holding itself way too full.